Hello, welcome to Science Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of June 9th. Let's start. First, we are going to discuss a paper published by Bernard Lantern in mBio on the role of bacterial biofilms in atherosclerotic plaques. Why is this paper so important? Not much is known about the role of bacteria in the atherosclerotic plaque formation. Authors show the presence of bacterial biofilms in the plaques. Authors also identify conditions that disrupt biofilms, leading to plaque destabilization. Authors analyzed 15 atherosclerotic explants by PCR and found 16S ribosomal RNA from bacteria in all of them. Density gradient gel electrophoresis on 5 of the plaques revealed the presence of common and uncommon 16S ribosomal RNA in all the 5 plaques. Confocal images of the eubacterial 16S ribosomal RNA gene sequence in the carotid arterial plaque showed the presence of gene as shown here in red. Authors show in vitro culture that the plaques could be disrupted in the presence of increasing amounts of iron. Where does the iron come from? Iron comes from transferrin in the blood, and in the presence of norepinephrine, plaques could be disrupted, releasing bacterial enzymes that might cause damage to the surrounding tissue. And released plaques could also block the arteries, leading to heart attack or stroke. Authors did not provide any data that this happens in vivo. Nevertheless, they conclusively show the presence of bacterial biofilms in the plaques. Next, let's look at another paper published by E.S. Wall in Science. Authors provide scientific proof for motor function revival after stroke. Why is this paper so important? Brain regenerative capability and recovery after stroke is limited. Treatment options like rehabilitation have had only minimal success. Authors show that sequential treatment of nerve growth promoting antibody therapy followed by rehabilitative training almost completely restored skilled forelimb functions after cortical strokes in adult rats. Here is the data to support their claim. Using photothrombosis, authors induced blood vessel blockade by multiple microthrombi, which destroyed more than 90% of the sensorimotor cortex of adult rats. Rats were then treated with intrathecal anti-NOGO-A or control antibody for 2 weeks. In addition, they trained the rats intensively in skilled forelimb reaching, either simultaneously with antibody application, that is parallel group, or during 2 weeks after antibody treatment, that is sequential group. When the growth enhancing anti-NOGO-A treatment was followed by the rehabilitative training, that is sequential group, animals improved their performance from day 16 post-stroke, and their skilled reaching abilities almost completely recovered to pre-stroke level, significantly better than all other groups. Authors looked at different sprouting patterns of corticospinal fibers from ipsilateral cortex of different groups. Their results suggest that the recovery of rat forelimb function after stroke in the sequential group originates from extensive and precise renervation of the stroke-denervated spinal hemichord by midline crossing fibers from intact motor cortex. Anti-NOGO parallel animals also showed growth, but nerve fibers had extensive branching and aberrant termination. Authors confirmed these results using various stroke and spinal cord injury models that are not shown here that relate midline crossing corticospinal fibers to functional outcome. So this paper provides experimental proof for sequential rehabilitation therapy following stroke for a better outcome. Thank you for watching Science Breakthroughs from Science Mission.